introduce Amanda Rigdon. She's a well-known fixture in the cannabis community from her support of the cannabis analytical industry through applications and education. Rigdon began working with the cannabis industry in 2011 and is currently an active member of the cannabis community as she continues to support sound science throughout the development of laboratory workflow solutions, one-on-one technical service, seminars like this one, and publications. Currently, Amanda holds the position of Chief Technical Officer at Emerald Scientific, a company that supports technical excellence in the cannabis industry through the introduction of the first series of proficiency tests, the first analytically focused conference, and the distribution of cannabis-specific laboratory supplies and equipment. Prior to joining Emerald Scientific, Amanda spent 9 years at ResTech developing applications, seminars, and technical literature for various industries, including the cannabis analytics industry. Prior to her time at ResTech, Amanda spent 2 years as an FDA GLP auditor. With that, I'd like to turn things over to Amanda.
All right. Thank you very much, Aaron. And thank you, everybody, for sticking around until the last talk. And thank you to all the previous presenters. It was all really interesting. So today, I'm going to talk about proficiency testing for the cannabis industry. What I've tried to do during this presentation is to kind of dig a little bit deeper into the details of proficiency testing. You know, right now, PTs are, you know, kind of a buzzword in the industry. They're being included in all of the regulations, and, you know, as Michelle pointed out, there are, you know, accrediting bodies, you know, legitimate ones and maybe some less legitimate ones that are, you know, getting into the game. So I think this is a good point to sort of give everybody a little bit of education. So when people talk about proficiency tests, you know, we'll know more about them than just what's on the surface. So today I'm going to be going over what is a proficiency test, talk about what PTs do and what they don't do, and go into the devilish details of proficiency testing. We'll see why they're devilish later on. Talk a little bit about interlaboratory comparisons, introduce some statistical concepts. I usually kind of make that face that lady's making when anybody mentions statistics to me. So don't worry, it's not going to be difficult or hopefully very boring. And then, you know, we'll talk about the path forward. So jumping right in, proficiency testing in a nutshell. You know, we have a PT provider or PT manufacturer that generates a sample with a known amount of analyte in it. For our purposes, let's say they generate a sample that they know somehow or another has 14% THC in it. Then that sample is, you know, relabeled with a blind label, so the amount in it is unknown to the lab recipients who get that sample. The labs analyze the sample, they report their results, and basically they get judged whether they pass or fail their proficiency test based on how close to the known value of the sample that they come in at for their results. I just want to point out the ISO documents under which labs and PT providers and PT manufacturers fall. So PT providers, they have to be ISO 17043 accredited. ISO 13528 and 5725, they're kind of companion documents to 17043. 13528 deals with statistical methodology for, you know, for PT providers in order to, you know, provide a good PT test. 5725 isn't quite as applicable to this industry because it deals mostly with standardized methods, and, you know, as Jeff and others very eloquently pointed out already, you know, we don't have standardized methods in this industry yet. You know, we're getting there, but we're not there yet. And laboratories, they have to abide by ISO 17025. Part of 17025 is participating in proficiency tests. So if we look at the list of, you know, what a successful laboratory must do, and it's not a comprehensive list, you know, as it applies to a proficiency test, the laboratory must get the right answer. And, you know, a PT does, it will tell regulators and it will tell the laboratories whether or not they got the right answer for that sample. You know, does a PT tell the laboratory whether or not they get the right answer every time? You know, it might give them a clue, but it doesn't say definitively whether or not they get the right answer every time. Can it track whether or not they, you know, do their ICOC, you know, their chain of custody, and follow all of their SOPs? You know, in a lot of cases, no. Does it ensure that they perform adequate QC? No. So, you know, a proficiency test does a lot of things, but there are a lot of other things that proficiency testing does not cover. So, you know, kind of one of the core concepts that I think to keep in mind is that proficiency tests are a part of an overall quality system or quality management system. So here we have a, you know, our little pyramid of quality. I just kind of came up with that, so, you know, don't reference it anywhere. This is kind of just my idea of the quality management system at a very high level. You know, at the base of the pyramid, we have our industry regulations. So, you know, in the case of cannabis industry, it's state regulations right now. Right above that, we have standard operating procedures for our laboratory. So the standard operating procedures are written to ensure that the laboratory meets industry regulations. And the people who write the SOPs in general, especially like in larger companies, are the internal quality and metrology folks as part of that department. And so those folks, you know, write the SOPs, but feeding into internal quality and metrology are all of your QC samples and your instrument qualification and all of the things, you know, that a laboratory needs to do to ensure that the data they produce each and every day is good data. And then kind of capping off that pyramid is regulatory enforcement. So, you know, at the top and the bottom of the pyramid, we have external influence. The middle, you know, is what you control yourself as a laboratory. Without either the top or the bottom of that pyramid, then, you know, there's a potential for quality to sort of suffer. You know, regulatory enforcement is pretty important because it's kind of, you know, the stick to prod laboratories along in ensuring good quality. You know, good quality is a lot more expensive than no quality, but it's very, very important. And so where PTs fall into this is that they're an outside check of your quality management system for a few different areas. You know, the PT can check whether or not your SOPs even, you know, address all of the industry regulations. They can also check out whether or not your quality, like your quality control, your QC folks, you know, stuff like that, are following the standard operating procedures. Because your SOPs may be written correctly in terms of you must run 3 QCs every batch, but whether or not the laboratory is actually doing that, you know, might be in question, and a PT might be able to show that. And then finally, PTs are, you know, kind of key in the interface between your laboratory and regulatory enforcement. You know, auditors always want to see your performance on your last PT and how you document that. So how does Emerald Scientific fit in? You know, we are a laboratory supply company. We are dedicated just to the cannabis industry, but, you know, we sell supplies and standards. So how is it that we fit into this whole proficiency testing arena in the cannabis industry? So if you look at the cannabis industry at a very high level, in general, regardless of state, across the industry, there's 7 common analytical methods. We have potency, we have microbiology testing, we have terpene testing, pesticide testing, residual solvent testing, mycotoxin testing, and heavy metals testing. That's 7 analytical methods. These 7 analytical methods potentially use 7 different analytical techniques. So it's not 49 combinations, but there's kind of a lot to know there and kind of be on top of the game in terms of knowing the market. In addition to those 7 methods and 7 techniques, there are potentially 30 different sets of regulations because of the different states that are legalizing or will become legal very, very soon. So basically, there's a lot to cover in terms of providing an applicable proficiency test. So we have our 7 methods, our 7 analytical techniques, a whole bunch of regulations, and I had to put this in because it's close to Christmas, a partridge in a pear tree. I hope somebody's laughing. It's hard to make jokes over a webinar. Anyway, and then the last thing to kind of point out about the cannabis industry is that it's an immature market. And when I say immature market, I don't mean generally that the people in it are immature or unprofessional. What I mean is the size of the market overall and the risk involved in the market. So the size of the market is pretty small as compared to, say, clinical diagnostics or toxicology or, food safety testing. It's a small market. And then because it's so young and so quick to change, the amount of risk involved in becoming involved with this market is much higher. And add in that it is not federally legal, this market prevents a lot of the established players from, dipping a toe into the market. So, there's just not a whole lot of support in terms of, outside input from a body that has a really good finger on the pulse of the cannabis market. So basically, at the very beginning, Emerald Scientific was founded kind of around the idea of facilitating sound science in the cannabis testing community. This picture is from February of 2014 at PitCon. It was kind of the beginning of Emerald Scientific. Emerald started in late 2013, but 2014 is when it really started taking off. Our CEO, Ken Snoke, and VP, Wes Burke, are in the green circle, and that's me in the blue. I was not part of Emerald Scientific at that point, but the important part of this picture is that the day that this picture was taken was the day that the Emerald test was born. So everybody in that picture, and also Frank Dorman, one of my colleagues who's behind the camera there, we were all sitting down talking, and we were talking about the needs of the cannabis industry. And one of the things that came up is that, cannabis laboratories need some way to make sure that they're actually performing to specification. At the time, laboratories, they were very understaffed. They were kind of novice. Laboratories have come a long way in just the last 3 years. But, the market really needed a proficiency test. So, that's when the Emerald test was born. And shortly thereafter, that's where the Emerald badge was born. So the Emerald badge was actually started because back in 2014, there were no regulations requiring people to participate in a proficiency test. So, the badge was kind of the carrot to go along with the stick. So it was kind of a way for laboratories to display, hey, you know what? We are doing something to elevate the quality of our science in our laboratory. And that's kind of how the Emerald badge was born. So really quick, I wanted to go through what the Emerald badge is. In short, it provides a marketing tool for scientifically sound laboratories. It's a way to improve the quality of science in the community. Laboratories have participated in multiple rounds of the Emerald test, and over time, they have improved in their performance. It is based on, established pass-fail criteria that is in ISO guidance. It's overseen by a third-party advisory panel, and it's part of the Emerald test that does have a proficiency test component. It's not overseen by Emerald Scientific. What the Emerald badge is not, it's not overseen by an ISO certified body, and it's not proof of successful completion of a PT. Now, the reason why I'm presenting this is because since recently, a lot of folks or a lot of regulatory bodies have required participation in a proficiency test. The regulators themselves, were kind of, there's been some confusion whether or not the badge is proof of the successful completion of a proficiency test. And the answer to that is no. Now, the reports that come with the interlaboratory comparison, where the badge comes from, are ISO accredited. They come from an ISO accredited body, and regulators can use that to assign pass-fail, but it's not a pass-fail. So I'm going to finish beating that dead horse. So before we go into a lot of the nitty-gritty details, I want to put some of the cannabis PT data that we're going to be looking at today into perspective. If we look at the cannabis market overall, and we look at regulations, and we look at what laboratories are expected to do, we find when we do a little bit of digging that there's very little specific guidance for cannabis laboratories in terms of, how they need to perform on a PT, which PTs they need to participate in, and stuff like that. What is my pass-fail criteria, basically? If we look at other similar industries, on the left, we have 42 CFR. That is CLIA, so that is the Clinical Diagnostic Laboratory Guidance. That is part of, the Code of Federal Regulations. And in there are actually pass-fail criteria for every proficiency test that these laboratories need to participate in. It's very well spelled out. In other industries, like the food safety industry, there are no pass-fail criteria. In fact, the guidelines for forensic labs and food testing labs don't say anywhere that a laboratory must participate in a proficiency test. Instead, they say that they need to be accredited. And usually they specify accrediting bodies like ASQ, ANAB, AOAC, or any of a host of other ones. So, the regulations handle it differently. But regardless of how the regulations handle it, there is a ton of guidance for industry available in other industries, not cannabis. So in the middle here, we have a standard guidance document that, auditors and laboratories and quality people have access to for free. And this is actually, it covers current good manufacturing practices, CGMPs. I figure I'd include this one because that term is kind of thrown around a lot in the cannabis industry right now, and a lot kind of goes into CGMPs. So, anybody who wants to make that claim, I recommend that they take a look at these guidance documents. The other thing that's not really talked about a lot is federal funding for the cannabis market. If we look at, any other market, forensics, food safety, environmental testing, not only have these laboratories had decades to get where they are in terms of, technological ability and expertise, but they've also been supported year after year after year with federal funding. For example, the numbers that I have here are just, a single year spend from the National Institute of Justice, USDA, and the EPA. The EPA's whopping $1.1 billion was from 2 years ago, I believe. Not all of it is to labs specifically, but they're budget was impenetrable. So, but, you know, it was all to protect the environment. The other numbers are specifically to laboratories. So, you know, there's millions of dollars of straight up federal grant money. That's not even including, you know, federal backed loans that Jess had mentioned earlier. So, you know, in order to do good science, there is a very large capital investment required. And the cannabis industry is the only one that I know of right now that expects laboratories to perform at this high of a level with, like, pretty much immediately. So, you know, only on venture capital. It's a very different model when you're working off venture capital versus when you're working off grant money. So, you know, I'm not making excuses for laboratories, but I do kind of want to put this data into perspective. So before we go over any data, let's talk a little bit about the hidden details of proficiency testing. You know, when we all think about a proficiency test, we think that a PT provider sends us a PT sample and then we get our results back, right? It's kind of like a black box. But actually, what a PT provider is responsible for is something called a PT scheme. And a PT scheme is not just sending out a sample. A PT scheme, and you know, this falls under ISO 17043, involves determining your market needs, determining your true value, acceptance criteria, sending out instructions, and all of that kind of stuff. And all of these different steps have a lot of guidance and a lot that goes into it. And in order to, you know, make an effective proficiency testing scheme, you need to have some specific knowledge of not only the industry that you're making the test for, but, you know, also the specific tests themselves and the expectations of the customers of the laboratories. So, you know, a lot goes into making a proficiency testing scheme. In addition to proficiency testing schemes, or kind of part of the proficiency testing scheme, you know, as a laboratory, I have received my sample and I reported my result and I get my pass-fail back. And, you know, it's as simple as that. But it's really not that simple. You know, first off, laboratories are supposed to follow rules in order to, you know, successfully say that they successfully participated in the PT, whether or not they got a passing score. In addition to those rules for the laboratories, you know, there has to be some sort of acceptance criteria, the ruler by which we measure pass versus fail for laboratories. And, you know, acceptance criteria are kind of the core of every proficiency test. You know, are we going to measure performance with a ruler, or are we going to measure it with a yardstick? You know, acceptance criteria is really important. If we set our acceptance criteria so wide that everybody passes, then the proficiency test doesn't really have any value to anybody. It doesn't have value to the laboratories, to the regulators, or, you know, to consumers whose safety these labs are protecting. You know, the converse is true as well. If everybody fails, it doesn't do anybody any good either. You know, so choosing correct and applicable acceptance criteria is not an easy job. One thing that I kind of want to point out here is that term Z-score that gets thrown around a lot, and it isn't get thrown around, but it gets mentioned a lot. So basically, your Z-score is your acceptance criteria, or a Z-score is directly related to acceptance criteria. So it's basically, you know, the maximum amount of variance that you can have as a laboratory in your results from the true value. And that's what Z is. A lot of people get Z kind of mixed up with other terms, but, you know, Z can be basically defined any way we want. So let's take a quick look at the general rules for proficiency tests. So, you know, that was the box kind of around the labs. Now, these rules are not onerous in the least. Laboratories must use routine methods and SOPs, at least for the proficiency tests that they're taking to get accreditation. If you're taking a proficiency test as, an internal check as a last step in method development, it's not a validated routine method yet, but you want to test it somehow, a proficiency test would be great for that. But if you are going to be reporting these results to an accrediting body, the routine methods have to be used. You can't use your best instrument to kind of give yourself a better value. And then the last two, no cheating, no discussing your results. You can't send your PT sample off to a third-party lab and basically have them do your homework for you. Darn it. My slide won't change. Oh, there we go. All right, so acceptance criteria. There's kind of two groups of general criteria for quantitative analyses. A quantitative analysis is basically any test that reports a number instead of just pass, fail. So on the left, we have absolute error. So basically, you need to hit this value plus or minus 10%, and that's it. The other way of assigning acceptance criteria that proficiency test providers use is standard deviation. Standard deviation is basically a method of saying this one is not like the others. There are lots of different ways to set acceptance criteria based on standard deviation. So, there's a big difference. Regardless of, which criteria you use, the criteria must be fit for purpose. And we'll talk more about that in a little bit. So here we are, the S word, statistics. So right here we have some data, our reported hexane values for this last round of the Emerald test from the interlaboratory comparison portion. The assigned value for hexane was 402 micrograms per gram. So, which one of these labs passes and which one fails? If we take a look at this table, it's just kind of a jumble of numbers. We might be able to throw out a couple because they're way off, but in general, there's no way we can tell, what's actually good and what's not. And that's kind of where statistics comes in. It's a really powerful tool for making sense out of large amounts of data. The data sets that come in with the PTs can be quite large. It tells you a lot about how laboratories are doing and, how basically, you should be looking at your data. Also, statistics does somewhat support unbiased evaluation as long as you do the same statistics every time. And the established statistical methods that are out there are widely accepted by regulators, and they're easily implemented with existing software. However, the other edge of the sword is they can be misused if they're not fit for purpose. And, kind of the mirror of one of the earlier points, these established methods are widely accepted by regulators. So, regulators immediately will, they might latch on to, some sort of acceptance criteria that they're used to from some other regulations. And, they can be easily implemented with existing software. So, as a PT provider, if you already have software set up to do evaluation of a proficiency test, in a industry that has standardized testing, it doesn't mean that that software is immediately transferable to the methods run in the cannabis industry. Without taking a really close look at the statistics that are used to determine, pass, fail, and these statistical concepts we'll be talking about, basically, you have a blindfold on. So this is kind of where we get to the interlaboratory comparison. So the ILC is kind of illustrated at the bottom of this slide. So we have our PT provider that provides the sample of unknown concentration to the laboratories. Instead of just getting a pass-fail, the aggregate data, like these 4 data points, go into an interlaboratory comparison data set. And that's really what the Emerald test is. That's the product of the Emerald test, is the interlaboratory comparison data set. And interlaboratory comparisons are extremely important in terms of determining whether or not, a certain laboratory is performing adequately. Because if you don't know how laboratories normally perform, how can you say anybody is doing good? And normally they're used for tests where no or very little historical data is available. That's definitely the case here. And, generally, at least an initial ILC is required to set acceptance criteria, but preferably an ongoing interlaboratory comparison should be performed because, especially in this industry, laboratories have been improving, so rapidly that, pass-fail criteria from 3 years ago probably won't be suitable, this year. So moving on, our first statistical concept, the consensus mean. So a consensus mean is kind of important in terms of whether or not a laboratory passes or fails their proficiency test. And to define the consensus mean, let's kind of use an example here. Here we have a dollar bill. The width of that dollar bill is 15.59 centimeters. If we give a bunch of people, a ruler, like sort of, the same ruler, and we have them measure it, we'll see that we get a set of 6 results, but not all of those results are exactly the same. The consensus mean is just the average of those 6 results. Basically, in a nutshell, that's what the consensus mean is. Now, if you notice, the consensus mean is not the same as the real value of the width of this $1 bill. And, the reason for this could be due to any number of things, and there's a lot of statistical ways, to decide, the proper consensus mean and how much bias there is in your measurement. So moving on to standard deviation. So let's take our initial example, our width of a dollar bill. So we have one group of people measure the dollar bill with a millimeter ruler, and another group of people measure it with an inch ruler. And here are our data sets for, the 18 different results. And if we take a look, if we plot these results, just the raw data, we can see that the measurements that we make with a millimeter ruler are a lot closer together than the measurements that we make with the inch ruler. And basically, this spread, this range, goes into defining our standard deviation. So standard deviation is a way of expressing how close together are your groups of results. So if we take a look at the calculation of standard deviation, we can calculate this relatively easily, not like that. We can do it with Excel. So if we want to calculate standard deviation very simply in Excel, this is what we do. I don't know anybody who would sit around with, a calculator and a pad of paper. So we calculate our standard deviations, and for the millimeter ruler, we have 0.113, and for our inch ruler, we have 0.317. So basically, what this tells us is that if we measure with a ruler marked with 1/8 inch increments, the results that, anybody gets will have 3 times more variability than measuring with a millimeter ruler. And this is kind of a really good parallel to draw with proficiency testing, in any industry. So, a ruler is a ruler, but you know, not all rulers are equal. So if we look at the bottom of the screen here, there's two instruments. They're triple quad mass spectrometers, and, you know, they're pretty much required for doing pesticide analyses. The one on the right came out, you know, 9 or 10 years ago at least, and, you know, it's a pretty good instrument, but if you look at the one on the left, that was brand new a couple of years ago, and the difference in sensitivity between those two instruments in some cases is a factor of about 100. So, you know, the measurements that you make on the instrument on the right may not be as accurate as the ones that somebody makes using the instrument on the left. So even if a laboratory says they're using MS, you know, it doesn't mean, you know, that they are making super accurate measurements. So our next concept that we're going to look at is normal distribution. And basically, if we take a plot of all of our results, basically the number of results versus, you know, the quantitative result itself, basically we'll see that we have a normal distribution in what everybody recognizes as a bell curve. The distribution should be centered around a consensus mean. You know, it can be skinny or it could be wide. And, you know, that's basically the spread of your analytical results. But the thing that we need to take home here is a normal distribution is bell-shaped. So if we combine standard deviation and normal distribution, we can see this like really cool, almost, you know, miracle. It's like the miracle of the Nautilus shell, right? The blue lines indicate one standard deviation away from the black line, the consensus mean. The red lines are 2 standard deviations away. And basically, this is the same thing that we were looking at before. It's a group of quantitative results. And if you have a bell curve like this, 90% of laboratories who made these measurements will produce results falling within 2 standard deviations from that consensus mean. And I think that's, you know, a really, really cool thing to think about is you can predict, you know, with 95% accuracy, you know, okay, well, a laboratory will produce results, you know, based on these data, a laboratory will produce results, you know, between this value and that value. And sometimes, depending on the ISO guidance that you use, a range of 2 standard deviations of the current data set is sometimes used for your acceptance criteria. So if we take a look at some real data, so this is, you know, the equivalent of that bell curve in the previous slide. It's just real data now, so, you know, it's a lot more messy. But if we take a look at it, it does kind of follow a bell curve. I have the consensus mean marked out here. And here's 2 standard deviations. So, you know, that looks pretty nice until we look at our x-axis. So basically, if we use 2 standard deviations as our acceptance criteria, that would result in 95% of laboratory results ranging from 5.2 to 7.8% of THC in a real-life sample that has an actual value of 6.5% THC. Is that good? Is that not good? I don't know. You know, it's not up to me to decide. But, you know, your pass-fail criteria, where you put those blue lines, is going to determine how far away from the actual value most laboratories will fall. So if we take a look at another data set for residual solvents, this is total xylene. Once again, here's our 2 standard deviations. You know, it looks very pretty. But if we look at our range, for example, with an actual 819 ppm amount of toluene, 2 standard deviation performance criteria, you know, basically allows for results ranging from 284 to almost 1,400 ppm of toluene. And so basically what this is saying is that, you know, the measurement, the allowable range, you know, will basically result in results that are not very accurate at all. And considering the fact that, you know, batches are passed or failed based on these data, it might be a little bit more important to be accurate here. And so that kind of brings us to our fit for purpose. Remember, several slides back, we were talking about, regardless of your acceptance criteria method, they have to be fit for purpose. And just using the simplest model of 2 standard deviations, which let me just say is that's basically recommended against in ISO 13528. But I'm just using this because it's the simplest method and kind of the easiest one to grasp. So for potency, if we ask whether or not these criteria are fit for purpose, how are we going to look at whether or not the criteria are fit for purpose? I decided, let's go ahead and look at the pharmaceutical industry. There aren't any established performance limits in regulations, and there's no variance list published because pharmaceutical companies don't want you to know what their variance is, most likely. But in general, I think consumer assumption is around plus or minus 10 to 20%. And if we see our 2 standard deviation range based on the interlaboratory comparison data from fall 2017, we're at plus or minus 20%. Is that good? Is that bad? It's all very subjective, and that's kind of one of the devilish details of proficiency testing. If we take a look at our residual solvents data, once again, there's a really good parallel to be drawn with the pharmaceutical industry. And if you take a look at consumer assumption for safety testing, I think in general, consumers don't think that there's a margin for error in safety testing. Like a laboratory tests it and it comes out with safe, then of course it's going to be safe. And that result is exactly right. But, there definitely is variance. So, a good allowable variance for this analysis, I have a lot of personal experience with it, is plus or minus 30%. So if we take a look at the 2 standard deviation range from the ILC data, we see that's a plus or minus 65% range. And basically, it's just because the data points are so far apart. Last but not least, pesticides. Once again, at safety testing, the consumer assumption is that, it's 0% error in the part of the labs. But if you look at food safety testing labs for pesticide residues, in general, laboratories, that are operating laboratories, they perform, anywhere plus or minus 40 to 50% is not uncommon. And, the reason for that is that pesticide testing is a very, very difficult test. We take a look at the range, that we got with 2 standard deviations. If we just, make that our acceptance criteria, that would result in a plus or minus 92% variance for myclobutanil. But, like I said, pesticide testing is very, very difficult, and laboratories are getting better every year. So fitness for purpose is really a balance. On one side, we need to balance consumer safety, right? We need to make sure that the safety tests that are being performed in laboratories are suitable to determine whether or not a product is safe for consumers. If they can't really quantify an analyte or, see a regulatory limit of an analyte, then there's really no point in a laboratory even performing that analysis. The other thing is, especially for the cannabis industry, industry legitimacy. The cannabis industry right now is under a microscope, and that microscope is only going to get stronger on us. It's kind of up to us to regulate ourselves. So in the interest of legitimacy, to make sure that we're policing ourselves right, we want to make sure that our laboratories are performing adequately. On the other side of the fit for purpose balance is cost to the laboratories. We don't want to overburden laboratories with a bunch of expensive proficiency tests every year. you know, that will basically reduce the participation rate, you know, from states or from accrediting bodies or from laboratories themselves. So, you know, really quickly, ways to determine fitness for purpose, you know, for acceptance criteria and just the statistics in general for a proficiency test is, you know, let's consider the importance of the test. You know, is this a test for just kind of quality, like cosmetic quality, you know, or is it a consumer safety test? Because if it's for consumer safety, then it's probably relatively more important than another test. And let's consider the end user's expectation and the ultimate use of the results. If growers are going to be using potency numbers, or extractors are going to be using potency numbers to dial in their process to basically change the concentration of their final products by 0.5%, then, you know, that 0.5% is within the variance of the laboratory. And that's a conversation that laboratories need to have, you know, with their customers, because all of this variance and statistics and all of that is kind of really hard to get your head around as a laboratory customer. You know, you just want the answer. So you can either sell your product, or you can make your necessary process changes, or any number of things. You don't want a qualified answer, but unfortunately from laboratories, you know, all the answers are qualified in one way or another. We can look for established specification limits for the finished product, right? If they're in the regulations, we can use criteria from different comparable tests, a bunch of other things. But, you know, I think in general, right now, a lot of it comes down to using common sense. You know, if we're allowing laboratories to, you know, have a 95% variance in their reported values, then, you know, that's probably not a good thing. Or, you know what, maybe it is, because right now laboratories are very good at identifying pesticides, but in terms of quantification, there's a little way to go, at least for some laboratories. But basically, you know, that's a discussion that needs to occur between laboratories, regulators, and consumers. So for steps forward, I mean, basically, you know, regulatory bodies, accrediting bodies need to understand the enormous challenges that are faced by the testing labs. You know, these are some brave folks. Let's continue to monitor and communicate results, you know, from the Emerald test. And hopefully, you know, we'll see even more increased participation from well-established accrediting bodies. You know, a lot of folks have stepped in this year, and I'm really glad to see that. And hopefully that will continue. So that's all I have, and I think I'm out of time anyway. So thank you very much, and I will take any questions that anyone has.